Welcome. I'm Gaia Trincucci, Deputy Editor at Journal of Experimental Medicine. Today, JM is hosting a conversation about immunology and women in science to celebrate the 2024 International Day of Immunology. It is with great pleasure that I'm going to introduce two exceptional immunologists and women in science, Dr. Arlene Sharpe and Dr. Kristen Pauken. Welcome. Dr. Arlene Sharpe is one of the world experts on T cells. She's the Collocatrons University Professor at Harvard University. She's also the chair of the Department of Immunology at Harvard Medical School. She's a member of the Department of Pathology at Brigham and Women's Hospital, and she's a member of the Broad Institute of MIT and Harvard. Dr. Sharpe is the leader of the Cancer Immunology Program at Dana-Farber Harvard Cancer Center, and she's the vice director of the Jean Lay Institute of Immunology and Inflammation in Harvard Medical School, Brigham and Women's Hospital, and Massachusetts General Hospital. Dr. Sharpe is also the co-leader of the Massachusetts Consortium of Pathogen Readiness, established in March 2020 by Harvard Medical School. She's an elected member of the National Academy of Science and National Academy of Medicine, and a fellow of the American Association of Cancer Research. And last but not least, I have the great pleasure of collaborating with Dr. Sharpe at JM, as she has been a member of the JM Academic Editor Board since 2019. Dr. Pauken is assistant professor at the Department of Immunology at the University of Texas MD Anderson Cancer Center. She received her PhD at the University of Minnesota in 2013 in the laboratory of Dr. Brian Fife, studying PD-1 mediated regulation of autoreactive CD4 positive T cells during type 1 diabetes. Dr. Pauken then moved to the University of Pennsylvania in the laboratory of Dr. John Werry and provided seminal contribution in understanding how PD-1 blockade impacted exhausted CD8 positive T cells. In 2016, Dr. Pauken then went on to study in the laboratory of Dr. Arlene Sharpe at Harvard Medical School, where she focused on understanding the mechanism of PD-1 mediated regulation of CD8 positive T cells in cancer. Welcome, and thank you for joining us today.
Thank you.
Thank you.
So I would like to start by our conversation today by asking what's your current research interest and what you are currently focusing on?
Well, my lab really focuses on co-inhibitory pathways and their roles in mediating homeostasis, tolerance, and defense against cancer, as well as infectious agents. We're looking at interactions with PD-1 and other co-inhibitory molecules to understand how they cooperate in different types of cells. And we've also developed genetic approaches to help us understand and identify new regulators of anti-tumor immunity and then discover their mechanisms of action.
What my lab is fundamentally interested in, the overall mission of my lab, is to really be able to expand the reach of immunotherapy strategies, including PD-1, to really improve on both efficacy and safety so that we can have more patients walking away cancer-free, but also free of other chronic diseases that may actually come up as a side effect of immunotherapy. So my lab is kind of split into two major halves. One is really focused on the efficacy problem of immunotherapy, and I'm bringing kind of my expertise as a basic cellular immunologist to really understand how the immunological starting material dictates response to immunotherapy, and then what types of combination therapies can be kind of rationally applied to be able to overcome the barriers that each individual has to a productive anti-tumor response. And then the other side of my lab is very interested in the immune-related adverse events that occur in the context of cancer immunotherapy. So many of these pathways that we block, including PD-1, are central regulators of immunological self-tolerance, or the ability of the immune system to know not to destroy self-tissues. And when we relax these pathways, a lot of people are actually just fine. And then others can go on to develop, either mild to severe to fatal immune-related adverse events that in many ways resemble autoimmune diseases. And we really fundamentally don't understand why some and not others, and really how those responses are different. And what we're trying to do is really understand how they're different from protective immune responses so that we could ultimately be able to dissociate them from the protective effects of immunotherapy, so we can really push hard on the immune system against cancer without kind of the collateral damage that can happen when we try to push the immune system against self-tissues. So that's what my lab does. It's kind of looking at both sides of the coin in the context of immunotherapy, protective anti-cancer responses as well as pathogenic immune responses that occur.
It's such an exciting time now, especially in this field, because also we're beginning to see agonistic reagents developing. The hope was that one could modulate these co-inhibitory pathways to take off brakes for cancer, but at the same time, could you use this immunoregulatory property to treat autoimmune diseases? And so we're embarking now on some of those studies as well. So it's just an exciting time to see how the basic understanding is translating to help patients with so many immune-mediated diseases.
Absolutely.
Yeah, absolutely. I have another question. So it is quite clear that you love immunology, and you've loved immunology since potentially the beginning of your training. So my question is, how did you decide that immunology was the focus of your research? So what sparked your interest in immunology back in the time?
So my graduate work was in virology. I did my PhD with Bernie Fields, and some of our work was really at the interface of immunology and virology. So that got me very fascinated in immunology. It was really at the early days of viral immunology. And in addition, I was very interested in pathogenesis of disease. And so that combination of pathogenesis of viral infections and viral immunity really pointed me toward immunology. And when I was a postdoc in Rudolf Janish's lab, the whole knockout technology was emerging. And I saw it as a wonderful way to begin to make altered hosts to understand the dialogue between the immune system and microbes is where I started.
So I actually started in the microbiology field as well. I like to joke that I caught the immunology bug, so to speak, in college. So when I went to college, I knew that I wanted to be in a biology-related field, ideally biomed, because I knew that there was always going to be just a tremendous amount of exciting research space related to human health and infectious disease. And so I wasn't sure what I wanted to do, but I went in that direction. And I was a microbiology major at Colorado State University. And one of our required classes was immunology. And I remember thinking how interesting it was and fascinating it was how the body dealt with infectious disease and how the whole immune system just really mobilizes. And, I went through innate immunity and I was like, oh, this is so cool. I went through the early kind of stages of adaptive immunity. And I just thought it was so interesting how exquisitely sensitive the immune system comes on and comes down and comes on and comes down. But what really captivated me and where I've spent the rest of my career and will spend the rest of my career is how quickly things get derailed when antigen doesn't go away. Like how quickly issues in the system start coming up when the pathogen's not cleared. And I was like, whoa, like this is the space that I want to be in and I really want to pursue. And so I worked in a chronic parasitology lab for a couple of years as an undergrad and then as a tech, really looking at fundamental mechanisms of how T cell dysfunction starts to occur and how the innate immune system instructs that in that kind of setting. And then when I went to graduate school, really just like this whole like box opened up of how different chronic antigens can be different for the immune system. And so seeing how chronic antigen in the context of autoimmunity was so different than infection, and then seeing how cancer is so different from the other two, and similarities and differences that kind of span all three of those fields. And it's just like how the body has to deal with this common thread of chronic antigen with completely different contexts has really been what's captivated me, and I think is an area that will only continue to grow and expand as our tools get better to study it.
That's so interesting because I think we started at different times. And so there were different kinds of tools that enabled different kinds of questions that I feel when I was starting in immunology, really the molecular biology tools were just becoming available to be able to go from phenomenology to really mechanistic understanding of the immune system. And so when I was starting, so many of the questions were, well, what is it that turns T cells on and what turns them off? And now here we are having just this incredible array of tools to be able to pursue questions in a more mechanistic way. It's really just a fantastic time.
Absolutely.
Yes, since we are talking also about women in science today, I am curious to know if during your training you had, women in science that you consider a model or someone that you would get inspiration from?
So it's interesting for me because I feel like part of why I went down a career in science sort of path actually stemmed from both a female and male role model kind of pair that I had when I was very, very, very young. So the initial kind of scientist that led me down the science path was actually my grandpa on my mom's side. So he is a nuclear thermal physicist at NASA and has been ever since before my mom was born. So obviously before I was born. And he literally got his job at NASA out of college and eventually went on to get a master's and a PhD and has spent his entire career just passionately loving what he does, like cannot contain himself, loves what he does. And seeing that as a very young child, I was like, wow, like I want that sort of excitement in my life. I want that sort of satisfaction that I get from just not being able to wait to get out of bed in the morning because the research that I'm doing is so exciting. He's now 87 and still is working. He still goes and he goes to conferences and gives talks. Like he cannot get enough of this. And that's what I wanted when I was really young. And what was interesting to me was that his inspiration, the reason he went to college, the reason he even pursued a field in higher education was actually his older sister. He saw his older sister in the 40s and early 50s going to college for a degree in math to become a mathematician and really excelling in a STEM science as a woman at that time. And his entire career trajectory, this life bending decision he made to go and pursue a degree in physics, was completely stemming from the inspiration that his older sister, my great aunt, provided to him. And seeing that as a young child, it was like, wow, like this woman inspired this man who has done so much in his life. I can do that too. I can eventually follow in those footsteps too. And so ever since I was 6, like this is what I've wanted for for myself.
Yeah. That's definitely a very early commitment.
So I became interested in science at a very young age, and I would say it's my elementary school teachers, most of whom were women. It inspired me. I was able, when I was young, to go to school and do little experiments before or after school that really tapped into my natural curiosity. But then I carried it home. And it was my parents who I think were just incredibly supportive that I set up these. I read about hydroponics and I set up these aquaria in our family room. And all I ever succeeded was growing mold. But they just let me like do this time after time, encouraged me to grow plants outside, which is probably why I like gardening to this day. But really the support, and the encouragement my parents really had that I could do whatever I wanted to do was that really strong support. But what I would say is that in addition to that, that along the way, I've had some amazing male mentors. When I was an undergrad, I worked in Jack Strominger's lab, and it was my first exposure to university science. I loved it so much, I couldn't believe they actually paid people to do this. It was so much fun. Bernie Fields was also an amazing mentor that he treated his lab like a family, and I think many of us who trained with him do the same thing. And then Rudy Yanish also was incredibly supportive in that way. And then Ramsey Cotran was my chair when I trained in pathology at the Brigham and also started faculty. And I learned from him what I always call him a mentor's mentor, because he had so much, gave me so much confidence. He had so much confidence in me. It gave me confidence in myself. And when I was going to start a family, I'll never forget, he said, I can't pretend to understand what you are about, how your life is about to change, but there are other women here who balance family and career, and I want you to talk with them and they will help you. And so I've just been so fortunate.
It's fantastic. Yeah, so regarding your training, as you said, of course, mentors are definitely one of the most important points. But I was wondering if during your training you had, some potential issue that could also inform any trainee now that are in immunology or in the field of biomedical science. And if you have any, tip or suggestion or what to do or what not to do during your training period.
Yeah, I think every person's journey that they go through in this field is so different. And I feel like, it's important to kind of take things as they're coming out at you and developing a really strong network of support and people who are around you who can really help you through the situations that you're kind of navigate. And it's hard to approach it as a one size fits all, because it's not. Like, there are certainly, I think, common issues that come up, for more people than others. But I think every person's experience is so unique, and that's really important to keep in mind. So a few of the things that have really helped me throughout my career is really developing both a strong peer network as well as a strong network of kind of more senior mentors, and really, being able to rely on both and extract information from both. I mean, people who have done it before versus people who are actively doing it now or have recently done it now. Like, it's really helpful to be able to kind of get in feedback on both sides of things. And one of the best pieces of advice I got was, make sure that you trust your gut, because it's important to get feedback from others, but people are going to be telling you what worked for them or what didn't work for them, and it might work for you or it might not work for you. And so being able to take all the information that you get and really come up with a solution that's workable for you, I think is really important. I will say, I think one of the biggest challenges that I felt like I was prepared for and then was in fact not at all prepared for was the transition to becoming a parent. And I feel really fortunate that I did it in Arlene's lab because she is somebody who really leads by example, and she's really somebody who provides a lot of support. I mean, she just talked about her mentors and how much support she got. And she absolutely paid that forward to everybody in her lab. And I think she probably saw what was coming for the rest of us, and we really didn't. It's like, oh well, I'm gonna be, I'm gonna be pregnant. I'm gonna give birth. I'm gonna have maternity leave. Thinking about how my pregnancy was gonna impact my experiments, because there's certain compounds that you can't work with during your pregnancy, and so you have to have support to be able to keep the experiments going. Thinking like, what on earth am I gonna do during my 3 months of maternity leave? And I definitely wasn't at all prepared for, like, okay, after your maternity leave ends and you go back to work, you're going to be the parent of a tiny infant, of which I had no experience with. Like, literally, I was leaving the hospital. I was like, I'm not qualified to do this. Like, how am I bringing this baby home? Like, I don't know what I'm doing. And that transition of like, okay, I didn't have a child and now I have a child was very jarring for me. And being able to figure out how to be a parent and then come back to work and then figure out how to balance being at work and being at home, and dealing with childcare and the cost of childcare and the unpredictability of childcare. And even if you have stable childcare, which I think during the pandemic we all struggled with, like, if your kid is sick, you've got to go pick them up. You can't leave them there. And so, like, always kind of being on call, and then just kind of figuring out how to balance the demands of being a mom with the demands of being a scientist. And it was really challenging. And being a mom immediately became so much of my identity. And like, my kids are hugely important to me. They're absolutely the center of my universe, and I want them to feel that. I want them to be like, my mom loves me so much, I'm the center of her universe. I don't want them to be like, oh yeah, I'm that side project that my mom has when she's not at work. Like, I don't want them to feel that way. And kind of figuring out like how to balance that was something that I was not at all prepared for. And I was definitely so glad to be in Arlene's lab when I was going through that transition, because she just fully got it. And she was so helpful for managing how to do that. And there was a couple of things that really have helped me. So one is my spouse, who's actually also a CD8 T cell cancer immunologist. And we were actually postdocs at the same time. We were grad students at the same time. And so we managed the transition to being parents together. And he was incredibly supportive. And like, we are absolutely 50-50 partners at home, like with the kids, with housework, with balancing everything. Like, he he is such a strong supporter of mine. It makes it so that I'm capable of doing what I'm doing. But then having Arlene just always understand, like, if I had to go, I had to go. And she really created like this lab culture where everyone kind of felt like they had a little bit of a safety net. Like, my kid's sick, I have to go. Can you split my cells? Or can you throw my stain in ICS or whatever? And her lab was always so supportive of that, and she was always so supportive of that, and it really made it so that that transition was something that I was able to do, and now it's just something that I do. Like, now it's something I have a lot of experience with and and balance every day, but it was definitely a very jarring transition.
Well, well, thank you, Kristen. Your words mean so much to me. I think I it took me a while. I was to develop this approach, and I think it's from my personal experience as well, that I was very fortunate to be a Lucille Markey Scholar that gave me the salary so I could afford childcare, as well as funding for a technician in the lab. And so when I started a family, I realized that I needed that support in order to be able to be, a mom at home and also to be productive in the lab. And I too have an incredibly supportive husband who shares, well, we share all sorts of things in life, but support each other and childcare and beyond, and, understand what time is all about in terms of being able to think about work-life balance and interactions. And so when I started a lab, it was clear to me that people need help, especially when they're starting families. And I think programs that are able to provide that technical support to be able to figure out ways to keep things going so people are involved intellectually, when people help each other and so forth to help people succeed. I think that's so important. And during your maternity leaves, you were inspirational. I don't know how it was during your second maternity leave. You wrote 3 papers during the pandemic, just amazing.
Thank you. And I will say, to kind of add on to what Arlene has said, watching how she helped us navigate the issues that we did in her lab, I am now trying to feed that forward into my own lab and create the sort of lab environment she had and be the sort of mentor that she was, because it meant so much to me. And so now I'm really trying to follow in the example that she set, to be able to be that for my trainees as well.
But I think, for me, mentoring, as you can tell, is near and dear to my heart. And I think everybody can be a mentor. We all are where we are because of mentors we've had. And so the graduate students are mentors for undergraduates. The postdocs are mentors for graduate students. And many of the technicians in my lab go on to graduate school. So the graduate students and postdocs are mentors for them as well. So I think, we're all role models at many levels. And I also benefit from peer mentors and as well as other mentors along the way. I think we're all lifelong learners. And, I feel very fortunate that I am part of a leadership group with other department chairs, and we get together and problem solve every week. And it's really an incredible group because I feel like we're all mentors for each other.
And that's amazing. I mean, I definitely feel that there is this, trickling down of good mentorship that you can see definitely in our conversation. And talking a little bit more about mentorship, so trainees can have a different, career path in mind. And I was wondering whether you welcome discussion about future career path with your trainees, and whether you sort of do a little bit of customized mentorship in terms of whether someone wants to go more into the route of becoming to having a career in academia versus someone that prefers to maybe leave the career in academia for industry or an editorial role, as myself, for example. So I'm curious about that.
I sort of view my lab as a family, and you want to figure out how to bring out the best in everyone and help them along their path. And I don't have any preconceived notions about what people are going to do. In fact, people may come in thinking they're going to have an academic career and change their mind along the way. And that's fine. It's very personal for everyone as they pick their paths along the way. And I want to be helpful. One of the ways I can be helpful is to introduce them to other people who have taken different career paths so they have opportunities to understand what each of these career paths is like. For our graduate students now, there are opportunities for them to understand different career paths with part of the graduate education, but also I've had people in my lab go into a variety of career paths, academic, industry, scientific writing, venture. And so there are different people that it's sort of a lab network that people talk with each other along the way.
For me, I think my kind of line in thinking here is very on par with Arlene and what Arlene said. And one of the things that I have become very cognizant to, during my time doing this is that I can't read people's minds, and I can't assume that I know what they want to do or what their goals are, or what they think that they need to get there. And so one of the things that we've started doing, based on advice that I had gotten from other senior mentors, was to have regular career development check-in meetings that are completely non-science. Like, as scientists, I think whenever we have a meeting, like the instinct is always to give the immediate project update, like, oh, this is the data that I have and this is the experiment that I have going on. But the purpose of these is strictly career development, and that's very vocalized to all of my staff in my lab. Like, it's a career development meeting, and that's what we're having. So for trainees, right now we do it quarterly, and then for non-trainee positions, we do it twice a year. And what we do at the beginning is everybody does an individual development plan so that they can actually communicate to me what are their long-term goals. And then together we can help come up with kind of more shorter term mediating goals to help them get there. And that way we're actively checking in regularly so that if things do start to change, we can modify the plan or bring in additional resources or additional connections to help best prepare them for where they eventually end up going. But I think meeting regularly about it and focusing on that topic has kind of given us the ability to kind of pivot more actively in real time. And in terms of getting people set up in the lab, I do think, when you're doing an academic position and someone's going to hire you out of an academic position, regardless of where you go, they expect certain things, right? They expect that you will have come in and independently led a project and ideally written a paper and figured out how to give an oral presentation on work in progress. And so all of those things are kind of a common thread across, all of the people in my lab who lead projects. But then kind of the sort of the nuance and the shaping and what each individual has kind of going on to help elevate them to where they want to go is more personalized and is more specific and totally flexible, because often when we start a position, 5 years later, when we leave that position, things have changed. Things in our lives have changed. Things in our careers have changed. And so I think checking in regularly and being able to pivot is really helpful for mentees.
Yeah, no, I agree. I think, I'm sorry. I think there are some kinds of life skills that wherever you take your path and pick your path are useful in terms of being able to communicate science, to an audience that's more specialized or to an audience that is a broader audience or even a lay audience, being able to write at different levels as well, to communicate in writing as well as orally. We wrote a lot of reviews and grants together. It was a lot of fun.
Yeah.
And, I think thinking about, different types of skills and being able to learn to be a mentor, to work productively with others in the lab, to be able to be an advisor to a graduate student or to a technician is all valuable for whatever path one takes. And then as one begins to think about it more, then one can sort of customize it.
Yes, I really appreciate this approach that is structured as well as flexible, and it's like a safe space where trainees can actually discuss, future development of their career, but also potential, issues that they can have, to actually reach that goal. So I think it's a wonderful way to approach mentorship and also to support trainees. So since we discussed about the mentorship, I would like to move a little bit more towards the transition from being a trainee to becoming independent. And of course, you did this at a different time. My question to you is, how hard was this transition? And if there were, like, again, points that you want to raise that could be important for trainees that now are thinking of moving into an independent position. Arlene?
So I think we did this at very different times in our careers. For me, because my husband was based in Boston, I was fortunate that Boston is a city that has many institutions. It's a great place for a two-career couple. So when I was looking for faculty positions, it was really focused in Boston. And so that was one aspect of it. I was fortunate in that I had this Lucille Markey Scholar Award that also provided me with funding during this transition period, and gave me flexibility to take risks, which was unusual but wonderful when I was starting my lab. I had done some work with writing grants as a postdoc and writing papers, of course, but learning how to write grants, writing an R01 was very challenging. And I was very lucky that Abul Abbas was my division chief, and he read all of my grants and gave me advice on them, far better that he did that than a study section. And, I pay it forward. I read other people's grants, they read mine. And, I think that's so important. So that support network really is something that I think has really been helpful. And I think I've sort of taken it to another level where all of the junior faculty in our department have mentoring committees. They choose 3 different people on the mentoring committee that they can go to and ask questions about writing grants, paper reviews of the grants that they've received, papers, and so forth, so that they don't feel like they're imposing on someone's time, but there's somebody there they can reach out to, and then I meet with them as well.
Fantastic. What about you, Kristen? Was it hard to do this transition, and also maybe to carve your, and your kind of niche away from Arlene's work, let's say? Or I know you collaborate a lot, but at the same
Yeah.
time.
So the transition was definitely challenging, and it's certainly something that I'm still navigating because I'm still fairly new to this. So, I think the biggest advice that I would give to people who are preparing to make the transition is you need to go somewhere where you're kind of scientifically and professionally feel supported, but it's also really important to account for your personal life as well. Like, is this a place where I can see myself, living and launching my career at the same time and having my kids if you have kids, like, because all of that really matters, and all of that counts. And so, you when you're making this decision of where you want to launch your lab, like, you really have to account for, like, that you're a whole person, and your family are whole people, and it's really important to keep that in mind. So, as I mentioned earlier, my significant other is also a scientist, and we launched at the same time. And ever since we were graduate students, we had always kind of heard of the daunting lore of the two-body problem. And it's so hard for an individual to launch and get a lab. Like, if there's 2 of you and you need to be in the same place, like, oh no. And we had kind of just gone through our training. We were both deeply passionate about what we did. We were like, we are just going to individually make each other, we are individually going to make ourselves as strong as we possibly can. And when we get there, we are going to shoot our shot, and we're just going to see what happens. And honestly, I do feel like the two body problem has gotten a little bit better. I feel like there are a lot of institutions that have become more sensitive to it. There's a lot of people who are more supportive of it. And it didn't end up being quite as daunting as I thought it would be. You know, we certainly had issues at some places, and you know, we were very vocal when we went on the job market that there was two of us, and that, you know, we both needed jobs in the same place, or it wasn't going to work. Like one of us wasn't going to go there and the other one was going to go somewhere else. It just wasn't going to work. And there were definitely places that were like, well, we don't want two CD8 T cell cancer immunologists to join our department. Like we want more diverse people. And it's like, well, like this is what we do. We're both CD8 T cell cancer immunologists. And so finding an institution that was not only supportive of us as individuals, but supportive of bringing us in and really facilitating our growth and development as two people who will not separate from each other was really, really important. And ultimately, we ended up coming to MD Anderson, and that has just been a place that's been tremendously supportive of us as again, individuals as well as together, and has been a place where we've been able to thrive. So we made the transition. I made the transition in September 2022. And similar to when I became a parent, I don't feel like I was at all prepared for exactly what that would entail. And I thought that I knew, right? Like I was like, oh, I'm gonna have to write grants, I'm gonna be directing people doing research, I'm gonna have to write an IACUC protocol. But until you actually live it, it's so hard to know what it actually entails to be a PI. And for probably my first 6 months, I always used to say my problem is twofold. I'm new to the role and I'm new to the institution. I'd never been here before. And so I really had no frame of reference. It's like, well, at my last institution, an IACUC protocol was submitted like this, and an IBC was submitted like this, and EHS looked like this. Like, you know, I had operated under those umbrellas, certainly at my previous institution, but I hadn't created that infrastructure. And it was incredible the amount of time it took me to actually build enough of a base in my lab so that someone else could go in and do a really simple experiment, like just thaw cells or inject cells into a mouse. And it was like, every lab that I've worked in, this was just there. Like this infrastructure existed. And I feel like took for granted that when I was a trainee. It's like I could go in, and after I had done my initial like institutional training, I could go and do an experiment the next day. And here, like I literally had to build the infrastructure, and that was tough. And then, you know, I feel like the other really challenging part is, you know, learning how to be really good at those aspects of my job that I don't have a lot of formal training in every day as I'm doing it. Like I used to be able to go in and just do an experiment or a couple experiments every week and just kind of regularly pump out data. And that was something that I was good at, and I knew exactly what to do, and I knew what to expect, and I knew how to troubleshoot. But when I started doing things here, like so much of my job, I didn't have that much formal training in, like trying to get my first IACUC protocol in. And like I spent so much time on it. And it was like, think of the number of experiments I could have done in Arlene's lab and the amount of time it took me to put together this IACUC protocol, which, you know, it's like, it's so different. Like how you spend your time is so different, and your responsibilities are so different. And then I think another real challenge when you first make the transition is that you have to really learn how to lead in that first couple of years. And it's not just how to lead other people's projects or how to lead the science in your lab, but how to lead a team of individuals in your lab, and the dynamic that you have one-on-one with each person versus the dynamics that people have between each other and you know, conflicts that come up, issues that come up, and figuring out, you know, what type of leader you want to be, and then how to actually effectively lead, I think is a real challenge for people at our stage. And again, like my institution has a lot of leadership courses and leadership tools to help with that, but there's, you know, there's something to be said for the experience of actually working through it, and experience is something that just takes time. So similarly, in terms of trying to like carve out my own niche in the field separate from what Arlene does, this is also a bit of a work in progress, because again, it's something that just takes time. So, you know, immediately kind of pivoting, like I need to be able to develop a research program. And the advice that I've gotten from many is it's important to start with what you know, because you need to be able to troubleshoot. like just getting cells to grow in addition in a mouse like is different at a different institution. Like, you know, I've had colleagues of mine who have just gone across the street in Boston have trouble growing their cells in their new incubator, and they're like, what's wrong with my incubator? And I'm like, I don't know what's wrong with your incubator. So, you know, it's hard to immediately just start in a new field because you have to be able to build a base, and you need to be able to rely on what you know. But you know, for me, Arlene has always been so supportive of what I've been doing. And she's always somebody who's really encouraged people to follow their passion. And so for her, she's like, you know, it's okay if it takes some time for you to figure out like what what is it that you're going to do and what is it that you're going to be. You just kind of need to take each step one day at a time to get yourself moving in that direction. And that's what I'm doing. And it's something that I'm really excited about and really happy with where I'm going, but it is a work in progress. And I think, you know, a message to other trainees making the transition is be okay with it being a work in progress.
Yeah, I fully agree. Sometimes we think about transition as an abrupt transition. So, okay, now you have your new lab, forget about what you're doing before. Okay, you use your experience, but at the same time, you are expected to have this new, completely new thing that you are developing far away from what you were doing before. But actually, this was your life for years and years and years. So it's also very hard to decide to detach completely. And I guess the role of mentors in this case is also very important because the transition is actually quite, you know, daunting for new PIs.
Yeah.
And I have a question for Arlene actually. So because you know, often junior PIs are asked what are issues and problems, but as an established PI, so is there anything that you still fear or like you have you know, or you feel like, okay, I reached the stage of my career in which I can completely relax, or what's the situation there? Because I feel that also established PI could have, you know, problems and things to solve, of course.
You know, I think part of it is time management, sort of building on what Kristen said, that there's demands on time. They're always changing in different ways in terms of different hats that I have and different roles that I have. You know, I love having the lab, the aha moment when you're the only one with, I have to live vicariously through data now. I don't do experiments with my own hands, but that aha moment never gets old. So I love seeing new data and discussing and planning new experiments. You know, so I think, you know, that's key to it. But I also have a number of roles being a department chair and other groups that I lead. So being able to have time management, and I think actually being a parent really helped me with that, because before I was a parent, I had the luxury to say, well, gee, I could work on this grant or I could work on this talk the night before it was due. And then suddenly it was like, well, I don't know if that time is going to be my time to work. And so I think I've learned over time to be much more organized with, you know, how I spend my time. In terms of, I'm a morning person, so I try to have my morning time be creative time in terms of writing or other things that I'm doing. And then, you know, meetings during the day and, you know, just organizing time in that way. I think, you know, transitions also kind of interviewing people for the lab, and you know, that's something that I had to learn when I was beginning. It's still something that I think is very important. I feel I have this incredible group of people. We create a supportive environment and they create a support network in the lab, that when we have somebody new who's coming, people interview people and they're very protective of the environment in the lab. And I listen very carefully to what everybody says because we want to maintain that environment. So I think, you know, challenging having an environment, you want to make sure that you keep it that way as well. And that also, you know, we're at a time when there are all these wonderful technologies, but it's expensive. So balancing writing grants, writing papers, data, other things, it's always a balance and sort of thinking how to organize one's time.
Fantastic. And moving to more of a general idea of women in science, you are both women in science. There are a lot of initiatives about women in science ongoing, much more awareness of women in science right now. So my question to you is whether all these initiatives and awareness and being vocal about women in science is changing the lab culture, and whether you have maybe some initiative that you want to discuss or that you are very, very, passionate about women in science, maybe in your institution or beyond that.
So this is an area where I feel like we've seen a lot of growth, and it has really just had a tremendous impact on, I think, women at all stages of their career, and really, creating the conversation and creating a safe space for the conversation to be had, I feel like has just made such a world of difference. One of the initiatives that I'm extremely excited about, I've participated in it this last year as a participant, but haven't actually participated in it as a facilitator yet, is the SITC Women in Cancer Immunotherapy Network, or WIN network. So a lot of our professional societies have really made a significant effort in terms of starting and sustaining the conversation, and just watching leaders in our field get together and have candid conversations about all sorts of different topics, because it's never just one thing, right? It's like so many different things that we're all kind of juggling at a time. And one of the things that I feel like has really been useful has been senior women in the field being able to have conversations about it out loud and talk about the issues that they have faced, or that we're still facing, and those sorts of things. So I think this is making a real difference. And one of the things that I think we're going to continue to see more of, and actually came up from the keynote at the SITC event that I participated in last fall from Dr. Liz Travis, who's actually someone from MD Anderson, is this concept of like, issues for women in science are not just women in science issues, right? Like, we can't just get better at dealing with all these issues by ourselves. We can't just get better at multitasking and time management and shouldering more. Like, we need to be able to bring men into the conversation. We need to be able to move some of the burden off of the shoulders of women, to bring in actual help. And I think that, rather than just like equipping us to be like super women at all things all the time, like how do we bring men in to be able to bring everybody together and help everybody rise, instead of doing it alone. And I think that's really a positive message.
I agree. I think that these initiatives really are making a difference. I think it's incredibly important to have safe spaces for women scientists to be able to talk candidly. We have a group called Female Leaders in Science at Harvard, that women faculty at all levels, there are different events, different programming, some that is less formal, some more formal. But for example, we had a dinner last fall that Joan Brugge hosted at her home, and it was wonderful. People were there. There were different icebreakers, and women, there were assistant, associate, full professors. And especially as we emerged from the pandemic, being able to interact with each other more, that I've stayed at Harvard because it's been a very special, supportive, collaborative environment for me. And for the young faculty, it's important for them to be able to begin to experience that. So I think that is really important. But in addition, Kristen, what you said about bringing men into the conversation, I can see that it's making a difference. That when I'm on a committee where we're planning a symposium or we're planning a seminar series, the men are very aware of trying to think of a balance of the speakers, so that there's a balance of men and women speakers that are going to be speaking at conferences, at symposia, in seminar series. And so it's wonderful to see that they're aware of this and that they're thinking about it and helping to create that balance.
That's great to hear. And, men in science are also, very well aware that women in science need support, and also, a space to and visibility. So I would like to conclude our conversation about basically going back to immunology, because we are here also to celebrate immunology. So my question to you is, what you think are the emerging topics now in immunology, and So what's you think is the next step, or something that it's kind of intriguing for you? Anything that you can, discuss about that would be great.
Yeah. I think it's such an exciting time in immunology now because we are getting more and more tools. I'm excited by the spatial technologies, the metabolomic technologies that now we can use to think about different tissue microenvironments. I think neuroimmunology is fascinating to think about the crosstalk between the immune system, the nervous system, and also cancer. The three together really, to understand that interaction, was really interesting. And then thinking about fields that really weren't traditionally thought of as fields where the immune system was involved in terms of thinking about metabolic diseases, aging, how diet changes events, I think are all really, fascinating areas, thinking about tissue homeostasis, resolution of inflammation and repair, and how all kinds of immune cells are there. And I think we're going to be able to develop a next layer of an integrative picture of homeostasis and understand why things work well when they work and how things go awry.
So to kind of expand on that a little bit, I do think that our rapid evolution of the tools and data science and systems approaches to immunology are really just being able to open so many doors for questions that we just otherwise weren't able to address. And I think one of the really exciting areas that we're making a lot of progress, or poised to make a lot of progress, is understanding human immunology. And what's incredible is that each person is their own walking case study. And being able to start to appreciate how the intersection between genetics, environment, other, chance elements of whatever it is that shapes who we are immunologically actually feeds back on our own health. So, when we think about a patient with cancer, how their infectious history has shaped things, how things like dietary factors and microbiome and, aspects of their environment have shaped things. And then when we start giving immunotherapy, all of those different contexts matter so much in terms of what happens with the anti-cancer response or whether an immune-related adverse event develops. And so really trying to understand, like, the cells of the immune system literally infiltrate every single organ of the body. They are absolutely positively everywhere. And they are exquisitely sensitive to their microenvironment. They're sensing what's there, they're sensing what's changing, they're responding to what's changing. And finally being able to have the tools to go into humans and understand how the immune system is shaping the human experience, human health, and how to best harness that to really have people living the healthiest lives that they can, I think is very exciting. Obviously, I'm on the immune-related adverse event side and really fundamentally interested in, why you can treat a huge cohort of patients with one immunomodulator, a PD-1 immunotherapy, and some people do just fine and other people don't, and why some people will develop myocarditis versus colitis versus dermatitis, like why it's so different. I think we finally have the tools to be able to start asking those questions in a really kind of sophisticated and high throughput way. And before it was just like such a black box. And so I think that that's really an exciting area that the field is moving.
I would agree. And I think human immunology, the tools are now allowing us to ask questions. One of the silver linings of the pandemic with COVID is that we're beginning to appreciate this variation in the immune system. Who knew that there were autoantibodies to interferons? I mean, it's fascinating, things that we're beginning to learn that are different. And, just thinking about the immune system compared to other systems, it's different if you say, gee, doc, how's my blood pressure today? How can you say, gee, doc, how's my immune system today? We've got to think about what we're learning to be able to answer that question. And I think we're getting there.
That's definitely fascinating. And with this last question, we conclude our discussion. And I'm really grateful that I had the opportunity to discuss with you today, and I'm sure this discussion will be very helpful for women in science, men in science, trainees, and beyond. Thank you very much for joining us today.
Thank you for inviting us.
I hope you enjoyed the conversation. JM has been committed to highlight women scientists and the work of women in science, so please do explore the content on our website. The JM editors wish you a fantastic International Day of Immunology, and thank you for listening.